Good afternoon, everybody. I want to thank the President for that lovely introduction, and of course Brent Johnson for inviting me to speak today. It's a wonderful pleasure to do this because this story is really about optics, ultrasensitive detection, lasers, of course, and also molecules, as well as biology. So we have a wonderful set of things to discuss for a little while, and I hope you find some of this interesting. So the roadmap for my talk, and where I'm going to actually use this pointer here to be able to let each group of people see a pointer, is to talk about super resolution microscopy based on single molecules. So that means I'll talk about origins briefly, and then explain super resolution with single molecules, although I know many of you know how this works already, giving a few examples. And due to time, I will also then talk about three-dimensional imaging and tracking with point spread function engineering. So that's one of the key things we've been working on, including using that idea to build a new three-dimensional microscope called Tilt 3D. I don't want to talk forever. You can see all these things that are grayed out because I deleted a lot to make sure I don't run over. But in any case, I hope that I won't run over anyway. So I have a big story to tell. Let's get started. So how did this all begin? How did this area begin? And in fact, way back 30 years ago, when I was at IBM Research, we achieved the detection of a signal that led us to single molecules, which was done in '89. I'll explain that a little bit. But it came from high-resolution laser spectroscopy, and it arose out of fundamental research on optical data storage ideas at IBM, at a company, at an industrial research environment. So that's a fascinating aspect of all of this, because back in those days at IBM Research down here in San Jose, the research goals involved either being famous for your science or for your technology. And that was the way you could succeed at IBM. And so we took that to heart, to be famous for our science, to try to understand. But one aspect of that was to explore a new application and the scientific fundamentals that underlie it. So the goal was to store bits of data in the optical properties of molecules using the color of the laser to record different bits in the same volume, because this could be done with a method called spectral hole burning that was invented in the 70s, where it allowed a sample to be changed in its optical spectrum to be able to store up to maybe 1,000 bits in one diffraction limited spot. So that idea, spectral hole burning, underlies this whole thing, but I don't want to explain hole burning in super detail, but I just want to step back and say this is because all of this was work done at one of these great industrial research labs of the time, where you could not only explore new technologies, but the fundamental science as well. So to explain this quickly, without a lot of detail, what I did to be able to turn this into single molecules was to ask and answer a question that was beyond the frontier of what was known at that time. So here's the idea. Imagine if I take pentacene molecules and I put them in a transparent crystal of paraterphenyl. That's a host material. And this is the optical absorption line at low temperatures of the pentacene molecules. So imagine cooling down the liquid helium temperatures, and this absorption line is the one, by the way, that we're going to make all those marks and bits in the frequency domain or in the color of the laser. All right, so that was the storage idea. But the fundamental question was, well, if you're going to do that, first of all, answer this. Is this optical spectrum smooth? Is it absolutely smooth? People thought it was a Gaussian shape, a smooth Gaussian shape. No one had seen any structure on that before writing any data. Okay, got the idea? We're talking about the background. What's there before you write data? And in fact, what we found in '87 was this amazing structure. So this was stunning, really. There's over now here a 4,000 megahertz range at 500 terahertz center frequency. You see all of these features. And blowing it up, you see the features. And just remember, this is not noise. This is not changing with time. You measure it once, and you measure it again, you see the same thing in one given volume. So we call this statistical fine structure because it's coming from the statistical effect that different numbers of molecules are present at each spectral frequency. And that, even though it has an average, it has a fluctuation around that average, which scales as the square root of the number of molecules, okay? So it was a square root of n effect, a spectral feature whose size scales as the square root of n. think about that, right? And if if you know of any other example, tell me. So this, of course, was maybe thought of as an esoteric effect at that time, and a background for optical storage, writing on top of this as well. But it turns out it led us to single molecules, because once you observe this, then you don't have to work any harder to get to the single molecule limit than square root of n. You don't have to work n times harder. If there's 1,000 molecules in resonance and you see this signal, its signal size is about square root of 1,000, or 30. So you only have to work 30 times harder to get to the single molecule limit. So it's a beautiful piece of spectroscopy put together with statistics, and that's what led us in 1989 to stay with this same kind of experiment, which was a transmission experiment. It was laser frequency modulation, okay, that Gary Bjorklund had proposed in 1980. It was using narrowband, single frequency lasers tuning with sidebands. You're measuring the conversion of a FM beam into an AM beam. And that's what the molecule did. It converted that FM signal into an amplitude modulated signal that we could detect. So here is a sketch of what this is supposed to look like. The FM signal is 2 copies of the molecule, and then here's a W shape, the derivative of that FM spectrum, because we were using another modulation to remove some other technical background. So this W is what you look for, and here you see W in a sample of pinnacene and paratorphenol, 1.4 Kelvin. So that's how this all got started. Absorption experiment. Now, a year later, another important absorption experiment occurred, and that was from Michel Horry in France, who also detected the absorption of a single molecule of pinnacene in paratorphenol, same sample and everything, but by detecting the emitted fluorescence. So fluorescence excitation it turned out to work beautifully. So at the beginning, this was a new arena, right? What's the most sensitive thing you could detect? Of course, people had done ions before, beautiful spectroscopy, and atoms, and electrons, and so on, and various traps. But here's a molecule that can connect to biology and all kinds of other stuff, including storage and things like that. But the storage idea never panned out. But the exciting thing here was the opening of being able to detect a single molecule with light. Now, I'm going to pass over details, but I just want to say that once you open up that new regime, surprises start occurring. That is, we saw immediately that these molecules were not just sitting there, but they had dynamics even at 1.4 Kelvin. They would blink on and off. They would move from one frequency to another due to low frequency excitations in the solid. All beautiful, fascinating science. Most people thought them to be problems. But we'll see what that means later. Let's see if this comes back. Optical switching we could do. We could move the molecules around in frequency space with light. So low temperature, of course, might have been thought to be esoteric, but at least it opened up this fascinating arena and got us all thinking about this. And so the whole field jumped to room temperature because people said, well, why can't we detect these at room temperature as well? Of course, the backgrounds are more of a serious problem, but if you are careful, then you can detect single molecules at room temperature by a whole variety of techniques. So I won't go through all of them, but I'll also just say that once we went to room temperature, surprises started appearing again. Single green fluorescent proteins, which is an absorber and a fluorescer that's used a lot in biological imaging, also blink on and off, even at room temperature, and also can be optically moved from one frequency to another. Again, thought to be, well, okay, these molecules are not so useful, but just keep that in the back of your mind. So rather than spending more time on details, I want to just set the stage for the room temperature experiments, okay? Because that's where everything has been since the mid-90s. While many methods work, what we're really doing now is we're pumping these molecules from an electronic ground state to an electronic excited state, maybe some vibrational sidebands. After some relaxation, then we're detecting the emitted fluorescence from the molecule as it relaxes back to vibrational levels of the ground state. So that's kind of the same thing that was going on at low temperature, but now we don't have the ultra-narrow absorption features anymore. Things are going to be broader, so it's easier to be in resonance and so on. And in fact, I'll explain why that's important in 1 second. You tend to pick molecules that don't have strong dark states because you want to be able to cycle the molecule, absorb, emit, get a lot of photons out of it, okay? So that's the way this works. Labels at room temperatures are basically laser dyes in some way. Here's a rhodamine, here's a cyanine dye, Psi3 in this case, and these green fluorescent proteins. All of these now work for single molecules. But when we do experiments at room temperature, think about how it has to be done to achieve the sensitivity that I'm talking about. Your laser is focused down, let's say, to the smallest spot possible if you're going to do confocal. And that smallest spot, as you know, is limited by diffraction. So in order to have only one molecule be pumped, you have to dilute the molecules. You have to place them farther apart than the width of this spot if you're going to detect only one at room temperature. That makes sense, right? And of course you can do this, or I wouldn't be here. And this works because, you know, we're using a fairly clean sample. We're using the shift in wavelength between the pumping light and the emitting light. That's tremendously helpful because you can then filter out any scattering from the pumping light that way. That's the Stokes shift. But at room temperature, you remember, in contrast to that low temperature regime. At low temperatures, features were super narrow. I could tune a single frequency around and pick out all the molecules differently. Now I don't have that power, so I have to dilute the molecules at room temperature. Okay? So, but nevertheless, this works. Here's a quick example of this. I'm showing you now some CHO cells, one CHO cell, Chinese hamster ovary cell. And this is a 12 by 12 micron field. There is one cell here. And when you look at these single molecules at room temperature now, they're no longer frozen, of course, and they're dancing all over. This is a transmembrane protein that's been labeled. And at room temperature, thermal effects make diffusion important in cells. And here you see these transmembrane proteins moving all around. Let me show it again because it's nice to watch and look at all the other things that are present in this little video from long ago. Here you can see one edge of the cell on the right, the other edge on the left. And these are all in a membrane. That's why they're all kind of in focus. But you see them turning off. So that's the photobleaching process. So photobleaching is a real thing at room temperature. You don't get an infinite number of photons. You get maybe 1 million. So anything you do, you must be conservative with those photons. We have to be careful how we use them, how we detect them, and we need to extract as much information as possible before they photobleach, right? And so on. So this particular regime has become a regime where a great deal of science can be extracted from those kind of experiments at room temperature because you can label an individual enzyme, watch it behave, label an individual motor, watch it move around in the cell, label DNA, see what kind of structures it produces, what kind of clusters or not clusters, and so on. This single molecule regime is still very powerful and interesting because we have many, many more to study. There are challenges for labels. We need great labels, and we need better ways to analyze the images and so on. But now let's switch to the Nobel Prize, okay? So that we go forward in time now to nearer to the present. And before I talk about the Nobel Prize specifically, let me ask you this puzzling question. What is the next best thing to being picked for a Nobel Prize? And you might say, oh, well, this guy's really crazy. Well, you know, maybe you have answers to this question for you. But my answer is being picked by Milhouse on The Simpsons. Yeah, so if you look here at the bottom, on the 26th of September 2010, this is 4 years before the Nobel Prize, episode 1, season 22. On The Simpsons, what's going on is that they're waking up in the middle of the night, and all the family's going, oh, why do we have to get up so early? So they're sort of dragging into the living room, plopping down on the couch in front of the TV. And why do we have to get up so early? And Homer says, because we got to watch the Nobel Prize announcements. And so they start watching the Nobel. Why are they watching the Nobel Prize announcements? Well, because they have a betting pool. Here's the betting pool. Here's the different family members. Here's the different prizes. And here's my name. You see? So you're really famous, you see. Really famous. And notice, you know, they're not so bad, right? Here's Ben Feringa, okay, who won last year. They picked Ben Feringa back in 2010. Okay, here's Bengt Holmstrom, who won in economics, okay, very recently. So these writers of The Simpsons, you have to have a little respect here. Anyway, So what are we doing, and why are we doing it? So let's just step back and remember there's a grand challenge we want to work on here. We want to observe this nanoscale molecular machinery that's at work inside living cells or complex systems. And here's one of these famous drawings from Good Cell. This happens to be a bacterial cell, a couple of microns long and a few hundred, let's say 500 or so nanometers across. So it's pretty small. And you'd like to image and see what all those little structures are inside, see what they're doing, okay? And, you know, looking more closely, any of these features that have been mentioned earlier are of interest, and they're all crowded and so on, but you'd like to see what's going on in there and try to understand it. And so your imaging requirements would be, of course, very high sensitivity, very high specificity if possible, living cells, and high spatial resolution. Well, you might know that electron microscopy has extremely high spatial resolution, and that's wonderful, but it's hard to use in living cells, and you don't have a way of specifically lighting up individual structures, so that when you look at the whole image, everything is there in grayscale, and it's a confusion to figure out what's what. There's a lot of effort to try to figure out how do you annotate, which structure, and so on. Whereas fluorescence is, as I've shown, extremely sensitive, single molecule sensitivity, very specific because we can attach those labels to a specific protein of interest. So the light will only be coming from just right adjacent to the proteins that you might want to see. And living cells can work. Okay, so, but the problem is, of course, with light, here's the size of that smallest possible focused laser spot. Only a few hundred nanometers in diameter, diffraction limited, of course, and it's really just too big to see what's going on inside this small structure. So that's one of the big driving forces for higher resolution. So we call this super resolution, but it's really circumventing the diffraction limit. Let me just show it a little bit differently. Here's a bacterial cell again. It's been labeled with a specific protein of interest has been fused to fluorescent proteins. So the light here is coming from those fluorescent proteins, labels. And to see what the structure really is, someone might think, all I have to do is to go purchase the most expensive microscope available. Here it is. Here's the most expensive microscope available. And of course, you don't see any structure. This is obviously because of the diffraction limit. Even though the labels themselves are only a few nanometers in size. So this is a fluorescent protein of 3 to 5 nanometers. Its spot appears really large due to Abbe's diffraction limit, stated in the late 1800s. And have been plaguing optical microscopy ever since, up until super resolution, lambda over 2 times the numerical aperture. So the point of super resolution, I just want to emphasize, is that you can change this image into that image, okay? That's why people are excited about this. These techniques jump a factor of 5 or 10 beyond the diffraction limit. So that's not a small effect, a factor of 5 or 10. In other fields, having an improvement in resolution of 20%, every year, or 30% or 50%, so forth, was a big step because you could always see more. Here we jump by a factor of 5 in a really relatively short time interval. And what that really means for us as scientists and optics experts and so on is how can we be sure that this huge jump and what we see in this new regime is always valid? That we haven't made a mistake because maybe we don't have a ground truth for something that hadn't been observed by light before. So that's one of our big challenges. Anyway, this prize was awarded to Eric Betzig and Stefan Hell and myself, and Stefan pursued a different technique, but the single molecule methods are more related to what Eric and I have done. So I'm going to talk about the single molecule one for right now. All right, so how does it really work? The key ideas are, first of all, we had to detect single molecules. So I already told you about that. That was done back in 1989. And we can image the fluorescence from them, either making a fancy 3D surface or look at a 2D camera image. And the key point first is to properly localize, to super localize each molecule by spreading out its light on multiple pixels of a detector, so that in cross-section, the different pixels are sampling different parts of the PSF itself, of the shape of the spot. Because the camera makes this measurement, okay, in each of its different pixels, then we can use a model function, let's say a Gaussian or an Airy function, to fit those measurements in order to get an estimate of the center of position of the molecule. Even though the width of this is diffraction limited, of course, the center position has an error function, okay, a statistical error, measurement by measurement by measurement, that's far smaller, whose standard deviation scales as the Abbe limit over the square root of the number of photons in resonance. So that's the key idea for localizing the molecules. This was not new, okay, to optical microscopy or single molecules or whatever. This was in science for many, many decades before, even all the way back to Heisenberg, who talked about localizing an electron by scattering photons off of it, and he knew you had to have square root of n, 1 over square root of n would control how well you would know the position of that electron. And so it's been around for quite a while, but applying it to these single molecules is what's, of course, more recent, because each of them is a tiny object, right? 2 or 3 nanometers in size. So that's great, and if you had 10 to the 4th photons, you could localize with 2 nanometers precision, a factor of 100 beyond the diffraction limit. But remember, this only works for isolated molecules, right? This only works when they're far apart. So what happens when they are close together and overlapping? Because what we're really trying to do is to achieve improved resolution, which means, in the formal sense, distinguishing molecules that are close together. How do we distinguish molecules that are close together? Much closer than the diffraction limit. And so that required idea number 2. I like to call it active control of emitting concentration, or sequential imaging. And this relies on the molecules having, in effect, 2 states. One state in which, when you shine light on it, it gives you fluorescence. Another state, even though you're shining light on it, it doesn't give you fluorescence, okay? That's the key idea, to have both of those possibilities, because then you can solve the problem in the following way. You have a structure that's been labeled with many fluorophores, and they're off right now. If you turn them all on at once, then this, of course, is the problem, right? All of the point spread functions overlap, and you just get the blurry image filled with fuzzballs. Whoops, sorry about that. So it's reminding me to stand up, but I'm already standing, so this is okay. Maybe you didn't see that message, I'm sorry. Okay, but when you want to explain this to your students, use this fuzzball idea, right? I like that, because the diffraction limit really means something looks like a fuzzball, and you can't make it be sharper, right? That's the key idea of diffraction. And so if they're all on, then they overlap. So the solution is, don't let them all be on at the same time, really. So you just turn a few on. And so that they are separated, and you can localize them, photo bleach them, and then turn more on. So you might remember that this is a random process. We are just improving the probability that they are going to be on, and so if you do that a little bit, due to randomness of Poisson statistics, a certain number will come on. But you don't know exactly which ones. Nevertheless, there's only a few on. So they provide samples of the structure. You can do it again and again, and then you take all those positions from the computer and show them all at the same time. So it's a reconstruction, really, okay? It's a reconstruction at the end, and you have to choose how you're going to display this data. But it really comes from single molecules sampling the positions on the structure that you care about, okay? So that's the key idea. And this idea appeared first in a meeting at the NIH in 2006. I heard Eric Betzig talk about it where he called it PALM. Soon after that, STORM appears from Zhuang, F-PALM. Paint, D-Storm, GSM, Blink, SOFI, all of these acronyms started appearing in the literature, a flood of acronyms. We demonstrated this using the switching of fluorescent proteins that I mentioned earlier, okay? But we didn't put an acronym in that paper. That's not smart, right? So to solve that problem now, I have another acronym to pile on just for fun, and it's mechanism independent single molecule active control microscopy, or SMACM. Huh. So anyway, the point here is a big part of this whole idea is this control of the molecules. How do you do that? And so as just a very brief flash, I just want to point out that's a fascinating problem for photochemistry, making of new molecules, chemists, designs, and creativity to try to make molecules that you can turn on and off. You want to make molecules that are fluorescent, and you can start with a dark precursor and convert that into the fluorescent one. That'll work. Or you can take molecules that go into dark states. And if you have the dark state lifetimes tuned properly, then you can shine light on a sample so that most of them are in the dark states, but only a few come back from the dark states and emit briefly. And that'll also give you a sparse image where there's a very small number of molecules emitting. So don't worry about all those mechanisms. But anyway, the idea here is clear. We're really measuring the different single molecules at different time points on a static structure. Okay, so they're giving us our information at different times. And so you can think of this as time domain multiplexing in a way, because remember the early single molecule stuff at the beginning was really frequency domain multiplexing. We had all these molecules at different frequencies, right? But now this is a different twist and works at room temperature for the purposes of producing images like this. So while there's hundreds of images out there now, I can't go through all of them. Just to give you a few, though, quick examples, because the basic idea is once you can see more, then you start discovering things. That's the real idea here. This is an axon, okay, which is present on all of our nerve cells. It's the long part that goes out to talk to another nerve cell far away. The axon has proteins in it, and if you label the spectrin proteins in the axon, you see that they form a banding pattern. A periodic banding pattern perpendicular to the long axis of the axon. This was not observed before, the optical measurements in super resolution. That's from the Zhuang lab. Here's a picture of what we can begin to see in the nucleus. Clusters of DNA locations that are particularly accessible to a labeling enzyme. Here's the Huntington proteins in a cell. It's a 2-micron scale bar here, and now this is a eukaryotic cell where these mutant Huntington proteins form these amazing fibrils that were not really reported before, these small fibrils that are all outside this really huge inclusion body. And in bacteria, remember bacteria are small, and so they profit from super resolution. We can now begin to peel back even the complexity that's in these bacteria. There are special structures that control signaling. There's special regions in the bacteria that are different from other regions. There's even ways in bacteria where you can divide asymmetrically, have 2 different daughter cells. And all of these fascinating problems are being attacked with super resolution fluorescence now. So great, that's all fine. So I've given you the basic ideas. And by the way, you ought to remember, maybe you have to explain to your children or your students or whatever sometime what this whole thing is, right, that I just described. So I want to, just for a moment, give you a nice little analogy. What is this super resolution by single molecules? Think of it like fireflies. Think of it like fireflies. Many people have seen fireflies. So you tell them this story. You say, well, you know, I want to see the branches of a tree at night. And of course, I can't see it, right? It's dark. I can't see the branches of the tree at night. So what I can do is I can place fireflies all along the branches, okay? And those now, if you just sit there and watch, just turn your cell phone on and take a movie, then you see that they will turn on randomly at different times, each one lighting up a location along the branches of the tree. So if you measure all those locations and show them all at the same time, you see the branches of the tree. That's the way it works. We just do it with single molecules. So anyway, it's not a perfect analogy, so don't push too far on it. All right. Now, back to optics, okay, for the last few minutes. 3 dimensions. What about 3 dimensions? What about the 3rd dimension? I've shown you 2-dimensional images of structures. And we want to see not only X, Y, but also Z, because these single molecules could provide us structures in 3D. And so we've been doing that. A number of people have been doing it with different techniques. You can do it with astigmatism and so on. But the real problem is that the conventional microscope with a clear pupil, so I'm showing now the key components of a standard wide field microscope, the clear pupil produces the standard point spread function, but this point spread function of a conventional microscope, as you move the molecule up and down in Z, then you see that, of course, it goes out of focus very quickly, and then in focus briefly, and out of focus briefly, and so on. It's not very good for determining Z. You can analyze this by Fisher information. Right at the focal plane, of course, it's bright and small, but its derivatives in the Z direction are 0. You have no good information about Z when the derivatives are 0. So the Fisher information gives you a way to talk about different point spread functions. And while there's many ways to do this, we've been doing it by changing what's in the pupil plane, or in the back focal plane, or the Fourier plane. Now, borrowing some wonderful ideas from Joe Goodman. So he was here earlier, and it's great to see him. What we've been doing is placing a phase pattern in the back focal plane that looks like this. This one came from Raphael Peaston in the University of Colorado. This phase pattern in the back focal plane changes a single emitter into a double spot on the camera. And importantly, if the object moves in the z direction, then these two lobes revolve around one another. So that for a molecule at a fixed position, let's say that position, oops, sorry, I'm not able to get it to stop in one position that I want, you have to imagine that the angle between those two spots encodes z. So by a calibration, then you know what z is, right? And so you have x and y, which would be the midpoint between these two lobes, and you have z from the angle of these two lobes. So it gives us a way to do the whole thing to get three-dimensional images. So I want to emphasize, we're not scanning anything when we do this experiment. I just moved the molecule up and down to show you what would happen if we have molecules at different z's. In a single experiment, everything is in a sense in one position, and so this is what you see on the camera. You see a bunch of pairs of spots, but you can see they have different tilts of those two lobes. And that's what gives you the z, right? And that means that we're imaging a whole thick slice of sample, really. The double helix point spread function is in focus for about 2 microns in z with high numerical aperture objectives. That's much larger than without the double helix, because you normally get only 500 nanometers or so in focus right near the focal plane. So remember, it works larger over a larger z range. Well, you know, this works for multicolors. This works because we're processing after the microscope. We're processing the image after it comes out of the microscope just by adding a 4F optical processing system with these phase masks. And so you can create three-dimensional images now of structures. I'm sure you'll believe me when I say there's a large number of point spread functions. So here's a bunch of them. They have different ranges. Notice here's one that works over 20 microns. My group has been creating new point spread functions in an optimal way recently by optimizing on the Fisher information, by letting the phase mask itself be the variable and saying I want to maximize Fisher information over a certain z range. And that's produced these tetrapod point spread functions. So remember, this is the image of a single molecule over many, many different z positions. And this has more information. The last thing I want to really say is, well, great, we've got all these point spread functions, we got all this business, but really, what about imaging cells? What about the really important thing of imaging those thick cells, these eukaryotic cells like we have in our body? Well, a lot of people have been applying light sheet methods to put a sheet of light through a thick cell so that you only illuminate that little thin pancake and not molecules that are above and below the focal plane. That's a fine idea. It's just that many of the light sheet designs previously have been very complicated. So we've just reported in the last month or two a new configuration, a new microscope that we call Tilt 3D, which is a tilted light sheet that also has point spread function engineering built into it. So the key point here is sort of illustrated in this little picture at the side. This is a side view of the microscope. And there's the focal plane. The dotted line is horizontal. But remember, we're illuminating with a tilted light sheet. But you might say, well, what the heck? Those that are above and below the focal plane are useless to you. No, that's not true. That's what these extra PSFs provide for you because they work over a longer Z range. I said 2 microns. So that means this molecule illuminated above the focal plane, I still record it on my camera, and it's going to tell me it's Z by the shape on the camera. Z comes from the point spread function, from the image on the camera, not from the position of the light sheet. And so this beautiful idea makes things far simpler because now I can just bring the light sheet in at a 10-degree angle. And easily get through the wall of a sample cell or a chamber or anything like that. I want to be able to image to all the way to the bottom of the cell, which this will easily do. So the illumination is super simple. It's just a stigmatic lens. It's just a cylindrical lens and just some optics to present that light sheet to the sample. So remember, some other designs work really hard to make a super thin light sheet or all kinds of things. So they have pairs of objectives that have to dip into the water and so on. All of that's gone here. It's just a standard cylindrical lens where we've used the tilt and the point spread functions to tell us what X, Y, and Z are. So this works. It works for mitochondria. Here's mitochondria in a cell. And here's a big structure, a 5 micron thick structure in the Z direction. The nuclear lamina It is a large sac that's right inside the nuclear membrane. And here it is in several different projections and reconstructions, coming from single molecule imaging, super resolution images, of course. Here's a cross-section that even shows you we have imaged a channel, an intralaminar channel. And here's, of course, the 3D image. So we're excited about having a way to sort of, marry the nice idea of a light sheet with the power of these point spread functions to operate over a long Z range to get three-dimensional images with super resolution. So I've talked too long, so I'm skipping a whole story about accuracy, how we spend our time trying to make sure that the positions of the molecules are accurate. If you want to ask me about that, I'm happy to explain it. Not just precise, but also accurate. And I just want to emphasize also that super resolution is not the only thing out there. These single molecules themselves are still useful in 3D. We use the 3D methods on them too, because you can use, and we can watch their motion. We can get dynamics from their motion. We can probe local environments and even local fields and nano antennas. And we can also use the light from molecules to look at photodynamics of complex proteins that are involved in photosynthesis. So all of this, of course, would not have been possible without my wonderful group of students and postdocs and my collaborators and all around the world and so on, and funding from the funding agencies. By the way, this picture here of the group right now, it's got a great spectroscopy joke in it. You see there's some gratings over here on this side and some light sources, and here's red, orange, yellow, green, blue, violet. So, and by the way, remember we look at one molecule at a time, but then look at another single molecule, another one, and anyway, but remember this is one guacamole. One molecule is one guacamole, right? Because it's 1 over avocado's number of moles. So, with the groans, thank you very much.